everyone. Thank you for coming. Welcome to the first open seminar this year that is powered by our WELCOME2 project. Today our special guest is Professor Kieran Tuohy. Kieran, thank you for coming. We met Kieran last November in Valencia during a Food Force network meeting, and when we invited him to come to Olsztyn, he would spontaneously said, yes, of course, I would love to. So thank you, Kieran, for keeping your word. Thank you for being with us. I'm not going to apologize for the weather because this is something you know, because Kieran came to us from the University of Leeds, where he chairs the Energy Metabolism and Microbiome Group at the School of Food Science and Nutrition. Before you were in Leeds, you worked for the Fondazione of Edmund Mach in Italy, in Trento. Here you led the Nutrigenomics and Nutrition Group. He also led the Department of Food Safety and Nutrition. Kieran also pursued his early career in University College Dublin, in University of Reading, University of Surrey, UK. Kieran is the editor-in-chief of the European Journal of Nutrition. His publication record is really impressive, 195 peer-reviewed international publications, book chapters in the areas of food chain microbiomes, nutrition, gut microbiology, and diet microbiome interactions. And today he will deliver the lecture on diet microbiome interactions in the gut, reducing the risk of metabolic disease. So Kieran, the floor is yours. Thank you, and go ahead.
Okay. Thank you very much, Ivana. And thank you all for inviting me to come here. It's a great pleasure to be in Austin. Only my second time in Poland. And I'm really enjoying it, especially the Irish summer you seem to be having. Okay, so today I'll be talking a little bit about my own research, but also in general about the gut microbiome and how diet can be used to impact on human health and disease by modulating the gut microbiome. So we'll talk about diet microbe interactions in the gut and especially how they might play a role in reducing cardiometabolic disease. So the gut microbiota is the collection of microorganisms that inhabit our intestinal tract. Now this microbiome, this collection of organisms changes geographically as we move down the intestinal tract from the stomach to the large intestine with different population sizes, different diversities of microorganisms. But generally we can say that it changes in geography along the length, but also transversely from the mucosal surface, the mucus layer, and the luminal contents, and also changes in time. So we're born sterile, we collect our microbiome from our mothers during birth, and then from the environment and our diet throughout life. By the age of 2, we have a narrow, tight microbiome, and in health that stays more or less the same until we get into our 6th decade of life and things start to change again. We know from human studies that all these chronic diseases present with a very different collection of microorganisms and their activities compared to healthy people. And we know mainly from animal studies that the gut microbiota are involved in all of these physiological processes that then link to pathways involved in regulating inflammation or pathways involved in regulating energy metabolism and storage in the body. And so it's not surprising then that we're making the link between what happens in the gut in terms of diet and microbe interactions and the regulation of inflammation and metabolic pathways that seem to be at the basis of obesity, cardiometabolic disease, but also autoimmune diseases, and more recently entering into some of the psychiatric conditions as well. So this is just a kind of a summary picture to highlight first that we have different collections of microorganisms in our intestines, and each individual here will have their own unique collection, partly derived from their mother, partly derived from their life experience, and that diet can change the relative abundance of these different bacteria. Especially fibers or prebiotics can support the growth of some organisms at the expense of others. And this ratio then governs the concentrations of metabolites that are absorbed from the colon and probably from the small intestine as well, the distal ileum. Metabolites like short-chain fatty acids, and I'll talk a bit more about these. Bile acids. The microbiome play a very important role in deconjugating bile acids at the end of the ileum and controlling reabsorption of bile acids, and also the conversion of primary bile acids into secondary and tertiary forms. And these different species of bile acids have different cell signaling potentials in different organs around the body. The microbiome also breaks down plant complex molecules, dietary fiber, as I said, but also polyphenols. About 95% of polyphenols we ingest, these are the color and flavor compounds in fruit and vegetables, are not absorbed in the stomach and small intestine, and in fact our bodies don't see them until the microorganisms chop them up into smaller molecules that then become bioavailable. So I'll talk about some of these different processes in more detail. Dietary fiber, so these are, we probably have all appreciate the important role dietary fiber has in human health and disease. If you look at most of the epidemiological studies, the one dietary factor that stands out as positively associated with reduced risk of chronic disease is usually always dietary fiber. Now, dietary fiber is mainly plant polysaccharides. It could be defined as a complex molecule that escapes digestion in the stomach and the small intestine and reaches the colon. Prebiotics, if you like, type of dietary fiber, are those that are fermented by the microorganisms in the colon and change their abundances and metabolite production. So these are two slides from George McFarlane. He was one of the chief players of dietary fiber in the 1980s and 90s. And he very nicely summarized what might happen in a traditional diet where, say, hunter-gatherers or people following a traditional Paleolithic diet, or even a pre-industrial diet in countries like China or Africa and India, where you consume 7 to maybe even 10 times the amount of dietary fiber that we do in the Western world. You see fermentation persisting right the way through the colon. And this means you have production of short-chain fatty acids in the proximal, transverse, and distal colon. And these metabolites are associated with beneficial activities. They regulate tight junction expression on the gut wall and control intestinal permeability. They're absorbed and regulate inflammation systemically. They regulate lipid metabolism in the liver as well, for example. On the other hand, on our Western diets, where we have much reduced dietary fiber, the microorganisms use the carbohydrate first and then switch their activities to try and derive energy from what's remaining in the diet. In our case, often amino acids. And the end products of amino acid fermentation, amines, indoles, ammonia, sulfides, and the nitrosyl compounds, many of these are DNA damaging. They're associated with tumor formation, they're cytotoxic, and they lead to intestinal permeability. And this intestinal permeability then leads to a chronic systemic inflammation that's associated with some of these chronic diseases. I mentioned bile acids earlier on. Well, bile acids trigger a number of very important cellular receptors that regulate things like inflammation, like GLP-1 secretion, insulin secretion from the pancreas, muscle contraction, inflammatory processes, vascular function, and also glucose and lipid and their own bile acid metabolism in the liver. So these are processes that are really key in regulating human metabolism and chronic disease risk. And what triggers these different receptors are different ratios and different species of bile acids. So the primary bile acids, cholic acid, chenodeoxycholic acid, they're conjugated or unconjugated forms. The secondary bile acids, things like lipocholic acid, deoxycholic acid, ursodeoxycholic acid, which is a tertiary bile acid, they all have different potentials to act as agonists or antagonists for these receptors. And so they can turn on and off these pathways that regulate energy metabolism and inflammation. This happens in the gut, but it also happens in the liver and systemically in different organs around the body. This is one example of an experiment from University College Cork from Susan Joyce's group, where she took 2 different bacterial genes for the enzyme bile salt hydrolase, which deconjugates bile acids in the distal small intestine. She cloned them into E. coli and then fed them to germ-free animals and compared it to conventional animals. So I don't expect you to know all these gene abbreviations, but just look at the change in color. Red is upregulated, blue is downregulated, and you can see germ-free animals, E. coli fed animals, the germ-free fed with E. coli with 1 bile salt hydrolase gene from 1 lactobacillus species, or another bile salt hydrolase gene from another lactobacillus species, and then compared to a conventional animal with their own microbiome. And you can see completely different gene expression profiles, both in the ileum and in the liver, just by 1 change in a bile salt hydrolase gene. So bile salt hydrolase have the potential to really impact on gene expression in the small intestine and also outside the intestinal tract. The other metabolic interaction between diets and microorganisms that's coming up as being important in human health and disease is the gut-brain axis. And this is generating a lot of interest in terms of irritable bowel syndrome, but also more chronic diseases like Alzheimer's, Parkinson's, depression, even schizophrenia are all showing different microbiomes and different concentrations of metabolites produced by the microbiome. I like this summary diagram because it highlights the 3 main communication channels between the gut and the brain, but also identifies that the microbiota are involved in regulating production of these neurotransmitters, especially gamma-aminobutyric acid and serotonin. About 80% of serotonin in the body is produced by the intestinal mucosa from tryptophan, and that pathway is regulated by microbial tryptophan metabolism. So how does all this relate to diet? We know what a healthy diet is, and we've known for a long time, probably from the days of Ansel Keys in the 1950s, who first defined the Mediterranean diet. And then you've had more recent iterations of this, the Lancet Eat diet, which is suggesting we reduce the consumption of meat, increase the consumption of fruit and vegetables and traditional foods, fermented foods from around the world. And if you look at some of the components of these healthy diets, especially down here at the heavy end of the pyramid, they're things that are rich in polyphenols, things that are rich in dietary fiber, fruit and vegetables. Fermented foods maybe in the middle, and at the top, where we're supposed to eat very little, you have the sweets, the refined carbohydrates, the highly processed foods, processed meats, for example. So if you like, you can break that complex diet down into a few components that impact on the microbiome. Probiotics, maybe from fermented foods, live microorganisms that when administered in an adequate amount confer a health benefit. So probiotics are in the foods you ingest. Polyphenols, as I mentioned, again 95% are not seen by the body really until they're broken down by the microbiome and the colon. And prebiotics, a take on dietary fiber that focuses on their interactions with the gut microbiome. So I'll give some examples of this now that we've used in Italy, but also in the UK. When I was in Italy, we studied the food chain microbiome associated with the local production. So we had apples, we had grape, but we'd also a traditional dairying, especially bringing cows from the valley up to the alpine pastures when they were sick or during the spring to put on weight to increase their health. And this had a big change, a big impact on the microbiome in the milk. So the microbiota of the milk was much different when the cows were up in pasture in the high alpine plains compared to when they were in house conditions and intensive production down in the valley. And so we were looking at this traditional chain and isolating microorganisms to see what they could do. And we found one strain that could produce about 10 times more gamma-aminobutyric acid than the other Lactobacillus brevis strains that were present. And so collaborating with University College Cork, We had our strain in purple, isolated from local cheese, the Cork strain in green, which was isolated from an infant in Ireland, and we fed it to high-fat fed mice compared it to a low-fat fed animal, a typical model of obesity and metabolic disease risk. Now, we didn't see much change in body weight, but we did see a big difference in how that weight was partitioned between muscle and fat. And in terms of cardiometabolic disease risk, that's very important because there's a much higher inflammatory output from fat than there is from muscle, and muscle burns energy rather than storing it. We saw a difference in microbiome. So these are the lean animals. These are the high-fat fed animals, and the probiotic fed high-fat animals somewhere in the middle. In terms of insulin homeostasis and cholesterol homeostasis, both probiotic strains reduced these markers of metabolic disease risk. We saw higher levels of gamma-aminobutyric acid in the adipose tissue and in the small intestinal contents of both these probiotic fed animal groups, and this was associated with an improvement in stress markers. So they carried out a test where they put animals on a forced swim test. They put on a platform, then you take the platform away, and you measure how quick the animal swims to the side. If they're depressed, they're a little bit more slow than animals that are less anxious. And so the probiotic from cheese and that from infant showed an improvement in these stress markers, showing that GABA production could really impact on the stress in this laboratory animal. Of course, we all know that probiotics have faced many challenges translating this data from perfect animal experiments into the human situation. So laboratory animals are very genetically closely related. They're housed under identical conditions. Their diet is very controlled, and their basal diet is very poor in nutrients. So normally you add anything to that diet and you see an improvement. Humans, on the other hand, are very genetically diverse. They eat and drink what they want. They have a very different environmental exposure than laboratory animals. And so it's not surprising that some of these very subtle nutrition experiments are not translated that easily into humans. Another challenge is that we've always focused on the colon as a probable site of action for probiotics. But when you think the colon has numbers of 10 to the power of 11, so 100 million organisms per gram of contents, the 10 to the power of 9 organisms you drink in a probiotic will make up less than 1%. So it's very unlikely that they're going to impact on the colon. However, in the small intestine, you have numbers about 10 to the power of 7 organisms or colony forming units per mil of contents, mainly of the facultative anaerobes as well. So things like lactobacillus and streptococci that we use also as probiotics. So when you consume a probiotic drink of 10 to the power of 9, that strain then likely becomes the dominant strain in your small intestine. And as I mentioned earlier on, the small intestine is very important for bile acid metabolism. It's where about 90% of bile acids are reabsorbed and shipped back to the liver to have a cell signaling potential. This small intestinal target for the microbiome has been demonstrated by others, by Freddie Troost, where he used complex endoscopy methods to sample the small intestine after delivery of 3 different probiotic strains or a placebo, and he could see differences in gene expression like that animal study I showed a bit earlier on. In University of Reading, as part of the Kabbalah Diet and Health JPI project that we led in Fondazione Mac, Reading, we're a collaborator, we did a dietary intervention where we fed oats, apples, a probiotic with a cornflake control or cornflakes as a placebo to healthy individuals, 12 people per group over an 8-week period, and we gave them a fatty meal challenge before and after to see how changing the microbiome might impact on postprandial bile acid levels. We didn't see much change in the apples or the oats or the control, but when we fed the probiotic strain, both in the acute setting, so immediately after feeding a probiotic, or after 8 weeks of continuous feeding a probiotic, we can see a very big change in the profile and concentrations of bile acids in the blood immediately after ingestion. So these are the kinetic curves for different bile acids, mainly unconjugated, secondary bile acids, and hydrophobic species being much higher, significantly higher when the probiotics is present compared to the other treatments. Moving on to polyphenols, as I said, about 95% of these compounds, these colors and flavors that we find in fruit and veg, are made up of very, very complex aromatic compounds. And basically these are not absorbed in the stomach and small intestine to any great extent, and they require to be broken down into smaller molecules before they can become biologically available for absorption. Now this makes sense if you think of dietary advice to increase fruit and vegetable intake. On the one side we have a trend in the general media to try and identify superfoods, blueberries or avocados or whatever the flavor is of the month. But that doesn't make much ecological sense because there are very few animal species that are evolved to consume just one plant. Maybe pandas are probably, or koalas are the only example. Humans are omnivores. We're expected to eat a diverse range of foods. And each plant has hundreds of thousands, maybe tens of thousands of these different forms of these complex molecules. But many of them are broken down into a smaller range of about 30 or 40 small phenolic acids. And it's turning out that these more common derivatives are the ones associated with regulating energy pathways and inflammatory pathways associated with chronic disease risk in humans, the same as short-chain fatty acids and bile acids. So if you like, the microbiome is acting like a funnel, taking very complex structures from plants and producing a smaller range of biologically active metabolites that then can impact on different pathways around the body. In Fondazione Mac, when we were there, these were in vitro models of the gut that I learned at the University of Reading when I was there. We applied them to look at how fiber and polyphenol contents, composition of different complex plant molecules could impact on microbiome in terms of ratios of bacteria present, but also in terms of the metabolites released. So using this in vitro model of the colon, we could see changes in beneficial bacteria, bifidobacteria, rosburia, and doria being stimulated by allurone, soluble wheat bran, an ultra-fine milled wheat bran fraction, or total wheat bran. On the other hand, organisms that we consider as harmful, such as E. coli, Parabacteroides, or Biophila, tended to decrease. So then did a human study in Fondazione Mac where we took overweight individuals, 80 people overweight or obese, and fed them either a placebo or this allurone, wheat allurone supplemented food products. And we made these breakfast cereals were made in Switzerland, but we made the breads ourselves in a local bakery, which has quite some funny stories associated with it. And then we fed them to these two groups and looked at how it changed the microbiome, changed metabolite production, and also impacted on markers of cardiovascular disease risk. Now, unfortunately, we didn't see any improvement in obesity measures or glucose or insulin measures, but we did see a significant change in microbiota, an increase in bifidobacteria and lactobacillus. And we saw a very distinctive change in circulating metabolites associated with some of these small phenolic acids that are released when the microbiota break down complex polyphenols. Now in cereal fibers, the majority of polyphenols are actually intertwined with the fiber. And so to release those, you have to also break down the dietary fiber. So this was a very good example of how you can translate or release the beneficial compounds in complex plant material through the gut microbiome. This metabolomics was carried out by somebody you probably all know, Marinka Julashevska, who's a friend of some here. She worked with me in Fondazione Mac and was really the lead on much of the metabolomics work that was done in Fondazione Mac in terms of nutrition. This is just correlating the metabolite production with gut microbiota composition, and you can see this Lectospiraceae being one of the main drivers of releasing these phenolic acids from the fiber. More recently, collaborating with University of Ulster in Northern Ireland, University of Parma in Italy, we tried to combine ex vivo studies with in vitro models to try and link changes in microbiome with particular pathways of metabolite production. So Chris Gill in Northern Ireland has a cohort of ileostomy patients. These are people who have lost their colon. Maybe they had IBD or colon cancer, or many of them in Northern Ireland, motorcycle accidents. So they can be healthy, or they can be people who survived chronic disease. But they have a stoma at the end of their small intestine, so you can collect the intestinal contents. And that's what we did after feeding them cocoflavanols, one of the few polyphenols to have a recognized health claim by EFSA for reducing cardiovascular disease risk. And then we mixed the intestinal ileostomy fluid with a fecal microbiome in our in vitro models of the microbiota and looked at how that impacted on the bacteria. We see different bacteria being stimulated by the flavanols and others, including organisms like some of the Clostridia that we think of as harmful being reduced by these flavanols. And then the PhD student spent a lot of time trying to link the presence of particular bacteria with different breakdown pathways for the different polyphenols, including a new recognized pathway for valerolactone production. Going on to more whole foods, so of course we don't eat supplements like dietary fiber or flavon-triols, we eat whole foods. And whole foods are made up of fiber and polyphenols. So another experiment we did, this was part in Fondazione Mac in Italy and part with the University of Reading. We looked at how 2 apples a day could impact on cardiovascular disease risk in people with high cholesterol levels. And these apples were unique to that part of Trento. They're like a cooking apple, except they're sweet and edible. So they're called Renata Canada. So people use them for making strudel, but you can also eat them as an eating apple. And they've got about twice the level of polyphenols as normal apples that you find in the commercial varieties in the shop. So we fed a crossover design, so 8 weeks on the apple or 8 weeks on a controlled beverage, which is an apple flavored drink with very low or no polyphenols and fiber, and then looked at various measures of cardiovascular disease and microbiome. So this basically just summarizes what happens in terms of cardiovascular disease risk and diet. In terms of diet, when the people are on the apple, we see higher fiber intake, higher total flavonoids, flavanols, some of the sugars associated with apples, flavon-triols in particular were increased, and so was quercetin. So this was all as expected when you're supplementing with 2 apples a day with a high polyphenol food product. In terms of weight, cardiometabolic disease, we see a significant reduction in total cholesterol, LDL cholesterol, triglycerides, and ICAM, which is a marker, an adhesion molecule, a marker of cardiovascular disease risk. In the total population, these were significantly reduced. And in the women, we saw a much higher response. We also saw a reduction in fasting insulin and VCAM, which is another adhesion molecule associated with cardiovascular disease risk. In men, we tended to see a much lower response. And this is recognized in other studies as well, where we see that women in general respond better to dietary intervention than men. And we know it wasn't that we had good girls and bad boys, because we took the polyphenols in urine and they were actually eating the apples. On the other hand, plasma bile acid concentrations were different between men and women. So we tried to model some of these associations, and we found that people who had higher lithocholic acid also had higher cholesterol. Older women had higher cholesterol, so the older you were, the higher the cholesterol. And these are, the age is definitely well recognized. Lithocholic acid is a new observation. When people were eating the apples and were younger, you had a more significant change in cholesterol. And this was also true when they ate the apples and they had high levels of glycoursodeoxycholic acid, one of these tertiary bile acids in their blood. So bile acid metabolism, at least in the females where he had a strong effect, was explaining the changes in cholesterol in terms of dietary intervention with the apples. So for the final experiment, I'd just like to show you some data from a collaboration with Ben Gurion University in Israel, who have done a large dietary intervention called the Direct Plus in people at risk of metabolic disease. They're pre-diabetic people. So here they were focusing on doubling the amount of polyphenols in the diet, 3 times the polyphenols in the diet. They called it a green Mediterranean diet, but also reducing meat intake and supplementing it with plant-based protein. And one of the alternative plant-based proteins they focused on was something called duckweed or water lily, and many of us will know this as the tiny little green plant that grows on ponds. So I'm sure here in Poland, but in Ireland it's very common on still fresh water. These are whole plants. They're very small. So like whole plants, they're full of dietary fiber, polyphenols, but also these water lentils have been used in Southeast Asia for generations as a substitute for meat, and they're called eggs of the water because their amino acid profile is very similar to eggs, and it's quite high in terms of percentage of protein. So the team in Israel, but also collaborating with Harvard and Dresden, looked at different aspects of how this plant could impact on human nutrition. They did acute studies showing that it could act as a good alternative to animal proteins in terms of release of amino acids postprandially, but also, unsurprisingly, they found that you could get high vitamin B12 after ingestion of this plant. And this is strange because most plants, maybe outside the legumes, have very low concentrations of vitamin B12, and vegans in particular tend to supplement with vitamin B12 because they find it difficult to get it into their diet. Here, because of a symbiotic association with a bacterium, the bacteria produce vitamin B12 in this tiny plant, and so when you ingest it, you get the vitamin B12. Another advantage of this is that each one of these particles is a whole plant. So I'm sure, well, some of you have tried the green drink. Sometimes it's even grass shots, and it tastes horrible. Not only is it green, but it's really a bad taste of chlorophyll. Because these are whole plants, when they're dried, you can use it as a whole plant ingredient, and so there's basically no taste. If you mix it with strawberries, you get a strawberry flavor. If you mix it with banana, you get a banana flavor. And so they found it very easy to get this into food products. So the study they did in Israel was a large dietary intervention. over 6 months and then followed the people for another 12 months. They had about 100 people per group, all pre-diabetic, so overweight and tending towards insulin resistance. And they had 3 interventions: physical activity, so free gym membership for the duration, physical activity plus the Mediterranean diet, where they gave them walnuts and green tea to increase polyphenol intake by about 440 milligrams per day, which is doubling which is reaching the Mediterranean diet recommended intake. And then a green Mediterranean diet, where they used this duckweed plant as well as green tea and walnuts to bring the polyphenols up to 1,300 milligrams per day, so 3 times the amount that you find in a normal Mediterranean diet. And then they measured various markers of health and disease, and for that's only MAC, we did the microbiome and metabolomics, or some of the metabolomics. They measured the usual cardiovascular disease measures, but also cognitive function and brain imaging in the people at 6 months, and then later on at 12 and 18 months as well. They had good compliance and people finishing the study, about 90 people per group. In all groups you saw a significant reduction in body weight, but this was greater for the Mediterranean diet and the green Mediterranean diet in terms of overall body weight and waist circumference. And this pattern of a dose response, if you like, in terms of polyphenols was obvious in all the different measures. So in terms of hepatic fat, you saw a significant reduction, the greatest with the green Mediterranean diet. And when they did correlation analysis, this was strongly correlated with 2 microbial metabolites, one of them neurangenin, derived from polyphenols or present in the duckweed. In terms of visceral or fat storage and different fat depots around the body, visceral fat was significantly reduced. Again, the green Mediterranean diet showing the greatest effect. And when they correlated these changes with metabolites, total plasma polyphenols, hippuric acid, urolithin and urine were all significantly associated with a reduction in this visceral body fat. And visceral body fat is the harmful fat, if you like, because it's got a much higher inflammatory output compared to other fat deposits in the body. In terms of microbiome, they saw a significant change in microbiome, again, most exaggerated on the green Mediterranean diet after 6 months. And this was associated with changes in main fiber fermenters, Prevotella in particular, increasing in abundance, but also the metabolic pathways associated with amino acid metabolism. So basically it looked like the microbiome was changing their ability to break down amino acids or produce amino acids. And as I mentioned, this is very important in terms of neurotransmitter generation by the microbiota. In terms of bile acids, again, you can see the healthy dietary guidelines, physical exercise, Mediterranean diet and green Mediterranean diet, big changes in plasma bile acid profiles. And of course, these were related to the microbiome. When they looked at the overall change in cardiometabolic disease risk and tried to explain it by comparing it with changes they saw in the microbiome, they could see that the microbiome changes significantly predicted changes in leptin, in body weight, in blood pressure, in the insulin resistance and the lipid metabolisms, all being predicted by this microbiome change, with the biggest change being mediated by the green Mediterranean diet. So they did one last experiment, and Ivana, this is not chocolate, this is something else. They did a fecal microbiome transplant. So, so when the people lost most of their body weight after 6 months, so after the biggest effect of this dietary intervention, they took 30 people per the 3 different diets and freeze-dried one of their fecal samples and allocated it out into 100 capsules that they fed back to the same individual over the following 8 months and compared it to placebo. So each of these dietary interventions had those that received their good microbiome profile induced by their diet or a placebo. And they wanted to see how this would impact on weight rebound. One of the biggest problems with dieting is that sure, we can lose maybe 5 to 10% of our body weight fairly rapidly if we go on a radical diet. But as soon as you stop dieting, you put the weight back on and you put on an extra bit. This is called the yo-yo effect, and you end up worse than you were before you went on the diet. And the same thing happens in terms of insulin resistance and risk of type 2 diabetes. When the people were on their own healthy microbiome after the green Mediterranean diet, this prevented the weight rebound, so they were able to keep the weight off, and it stopped the return of the insulin resistance, which is basically the precursor for type 2 diabetes. Okay, so just in summary, I think we're getting a much clearer vision about how the microbiome regulates inflammatory and metabolic pathways that control chronic disease risk in all animal species, but also including in humans. We're getting more and more demonstrations that feeding probiotics, prebiotics, or polyphenols, or whole plant foods made up of some of these compounds, or diets made up of these foods, can change the microbiome. And we're beginning to get evidence in humans to show that this change can then regulate the inflammatory metabolic processes that are involved in cardiometabolic disease risk. So finally, I'd just like to thank and recognize all my collaborators in different universities around Europe, but also especially my old team in Fondazione MAC, Francesca Fava, Matthias Schulz, who did all the metagenomics, Marinka Julashevska, Tarantino, who was really leading the metabolomics, and Francesca, my wife, was the main one running the human nutrition studies. Okay, thank you.